Good afternoon. Welcome back to the second year zoology class. We are in SAQs from the immune system. Before that, in the endocrine system, one SAQ I didn't take. That is also important. That one is the water soluble hormonal mechanism. Clear? The hormonal mechanism. The hormones are mainly the two types are there. Hormonal mechanism. This one is the actually it is visible now. That is the next lesson immune system PDF. But hormonal mechanism is mainly depending upon the hormones. They are the first one is the water soluble hormones. Water soluble hormones are there, and the lipid soluble hormones means the insoluble and means they are the insoluble in water. Lipid soluble hormones, how they enter means they are the two the receptors are there. They are the membrane receptors and the cellular receptors will be there. Through the membrane receptors, water soluble is enter inside. The lipid soluble they enter directly. They goes to inside. Clear? This one is about the water soluble and the fat soluble or lipid soluble vitamins. Clear? The first how the water soluble hormones are enter means, for example, here I'm drawing the diagram. Here the receptors will be there. For example, which hormones are the water soluble hormones means they are mainly the ovaries are having the hormone is there that is called as a FSH hormone, follicle stimulating hormone. Here the follicle stimulating hormone is the water soluble hormone. How it is means they are mainly here it is for the entering of that. Here they are entering of that. Mainly the secondary messenger is required for this one. Here next, for example, this one is the ovarian membrane. Here ovaries are having the ovarian follicle membrane is there. How it is occur means here the receptors are there. These receptors are taken this one and here giving the response one is there. Here after the response, the generation of secondary messenger. Here generation of secondary messengers. Secondary messengers are formed like which secondary messenger? They are the cyclic AMP, or the calcium ions. Both of them are act as a secondary messengers. Means first the response is there. How it is formed? They are formed as a secondary messengers. Secondary messengers give the response and occur the biochemical reactions are occur. Here biochemical reactions are occur. The biochemical reactions are occur. After that, the physiological reactions are occur. Physiological reactions are occur. Like this. After lastly, they are formed as a ovarian growth is occur. Here ovarian growth is occur inside of this one. Here this is about the water soluble, the hormones. So how they are means the water soluble hormones are mainly they require the secondary messenger for the transporting purpose. They are having the secondary messenger. How it is means, for example, follicle stimulating hormone. They are the stimulates the ovaries, means the here it is the first ovarian membrane. Ovary membrane is there. That the hormone is enter and giving the response. The immediately the generation of the second messenger is formed. That is called as a cyclic AMP or the calcium ions. After that, the second response will occur. They occur the biochemical reactions. After that, form it as a physiological reactions. With that, the result the ovarian growth and development. Here, this one is about the first one, water soluble hormones are how they are enters and they function. Here, next second. Next, we are going to discuss about the second type, lipid soluble hormones. Lipid soluble. Second type, the lipid soluble hormones. Means, for example, here it is the uterine wall is there. Means the inside the which one the this uterine means estrogen hormone. Estrogen hormone is the steroid hormone, or also called as a lipid hormones. Clear? This lipid hormone, how they enter means they are directly enter inside of the cell. Before that, they are not directly enter inside of the cell. First, they are form it as a secondary messengers. They take the secondary messengers. Secondary messengers are given to the changes occur in the biochemical changes and the physiological changes. Then the function will occur. But in this, not like that. The receptors are there. These receptors are taken and here they are given to the inner side. And so what it is? Here it is the this one is uterine wall. Uterine wall. Hormone is mainly the which hormone? Estrogen hormone. Estrogen hormone. Here it is form it as a east of the receptor and this one and this one are combined and form it as a the hormone receptor complex. Hormone receptor complex. This hormone receptor complex directly they enter inside of the nucleus of the cell and they change that one and they change in the proteins means that directly they are having the biochemical changes. The before that secondary messengers, after that biochemical changes are there, biochemical reactions are occur, after that physiological reactions are occur. But in this not like that. First they are formed as a hormone receptor complex is formed that act on the RNA and DNA and they are formed as a here the uterine wall, uterine tissue development. Here uterine tissue development is occur. Tissue development is occur. Here like that. These are two mechanisms are there. One is the water soluble the hormone mechanism, hormonal mechanism, and the lipid soluble hormone mechanism. Here this with this. The last question I didn't explain before. That's why I taken this. Next, we continue with the immune system. In the immune system, the already we discuss about the B cells. The B cells are the mainly the lymphocytes. B lymphocytes are also called as a B cells. What is the function of the B cells means? They are mainly helpful for the B cells or B lymphocytes are helpful for the releasing of the antibodies against the antigens. Here antibodies means the army in our body. Army is called as a antibodies. The reverse to the antigens. Any microbes are enter in our body. For that, the reverse army is producing in our body. That army we call as a antibodies. Here the B cells we discuss. Here the B cells are mainly they are formed as a these B cells are they are formed as a they activated they are formed as a two types. They are called as already the plasma cells Some are form it as a plasma cells and some are form it as a memory cells. Here, the what about the plasma cells means the plasma cells are every time they are producing the antibodies or they are releasing of the antibodies. Antibodies are released by this one only. What about the memory cells means they store the information about the particular antigen. Which antigen is enter in our body? For that, the information is stored inside of our body long time. Clear? This one is about the memory cell. Clear? Next we are going to discuss about the one more important the question that is called as the immunoglobulins. The visible na that diagram is visible na that only the immunoglobulins. The see here. The what about the immunoglobulins? See this. Immunoglobulins are also called as the antibodies. Here, the already B cells are producing the antibodies now. What is the structure of that antibodies are also called as the antibodies. Another name is called as the immunoglobulins. Here, the what it is means simply they are Y-shaped structure. Here, see this one. Antibodies produced by B lymphocytes are also called as B cells. Before only we discussed. The B cells are main function of the B cell is the producing of the antibodies. These antibodies fight with the antigens. Clear? Next, when the pathogens are enter in our body, when any microbes enter in our body, microbes are considered as a antigens. Antigens are enter in our body immediately. The antibodies are produced by the B lymphocytes are also called as a B cells. Clear? The antibodies are antigen specific. Antigens are enter in our body immediately. That particular antibody is released. That's why it is a specific. The antibodies are antigen specific. They are next. Based on the mobility, based on the movement, there are two types are there. Antibodies are two types are there. What are they means? Here I am writing here. See this one. Here the two types of antibodies are present. They are called as a surface antibodies and the circulating antibodies. First one is the circulating antibodies. Circulating antibodies. Surface antibodies. Surface antibodies. Antibodies depending upon the mobility, depending upon the movement, they are two types are there. Circulating means freely circulating in the blood and lymph. That's why we call it as a circulating antibodies. What about the surface? Just they are but attached to the cell. Surface of the cell, they are present. They attach to the cell. That's why we call it as a surface antibodies. Here, depending upon the movement or mobility, there are two types of antibodies are there. They are the circulating antibodies and the surface antibodies. Here, next is the circulating. What about the circulating antibodies? Circulating antibodies present inside of the already I said where it is present inside of the blood or lymph. They are freely moving. That's why they are present in the where they are present blood and inside of the lymph they are present. That only they given here. The next is the what about the surface antibodies? Surface antibodies are present in the surface of B cells. Before only we discussed that. I'll draw the diagram. See, observe here diagram. The like this. The B cells are yesterday I draw the diagram. B cells are like this. They are above the B cells. The Y-shaped structures are present. These structures are called as the antibodies. Here, like this, they are the some B cells are having the surface antibodies are present. Means they attach to the surface of the cell that we call as a surface antibody. See, here the surface antibodies present in the surface of the B cells and also present in the memory cells. In the memory cells also they are present here. Next is the what is the structure we are going to discuss. Structure of the immunoglobulin. See here, I am drawing the diagram. This one is an important diagram. Compulsory you draw the diagram. This diagram, see how I am drawing this diagram. See, The Y-shaped, simply would write the two long chains are there. I will explain afterwards. First, observe the diagram. They are having the, actually it is looking like a Y shape, and this is having the two long chains are present. This one is a long chains are present. Polypeptide chains. These are the polypeptide chains. Two are present. One pair is called as a, or two chains are called as a heavy chains. Here, observe here, this one, this center is in a, that is called as a heavy chains. Clear? Next is a, again two are there. They are called as a short chains. See here, this one is the short chain. That is called as a light chains. Short, it means that's why it is called as a short chains are present. These two are the light chains are also called as a short chains. These are the long chains are present. Next here in this mainly the between them the disulfide bonds are present. Between the heavy chains the disulfide bond is present. Here like this. Here also see this one. The disulfide bonds are present, and this is called as a heavy chain. This is called as a heavy chain. Heavy chain, and this one is called as a smaller one is called as a light chain. Light chain. Between them, the bonds are there. They are called as a. This one is called as a disulfide bonds. Disulfide between the two heavy chains, and between the heavy chain and the light chains, the bonds are there that we call as a disulfide bonds. And here this place we call as a, this the upper Y shaped is there, that is called as a FAB, means the FAB end means antigen binding site, means this one is a one paratope. This one is a, this side is a one paratope, and this side is also called as a one paratope. They are having they are for useful for the binding of the antigen. That's why we call as a FAB, fragment of antigen binding. Next is here the FC are there. Here, what is the FC means? The fragment of the cell binding. Here, that the one fragment is attached to the, for example, draw the diagram before only now. This one is a B cell. B cell is having the, like this, the antibody is present. Above the B cell, antibody is present. This is binding with the B cell. That's why this is called as the FC, means fragment of cell binding site. Next is the, above that one, FAB, fragment of antigen binding site. Like this, it is having the, which shape it is having? The Y shape is there here. This one is a one paratope. This one is a one paratope. Between them, the disulfide bands are there. This is the FC end. This one is called as a FAB end. Clear? Next. Observe the structure. The structure antibody is which shape? Y shape molecule. And how many polypeptide chains are there? Four polypeptide chains are there. What are they? There two are the long chains, or two are the short or light chains. That's why the four polypeptide chains are present. This is a Y shape. These are having the two polypeptide chains. Two are the heavy chains, two are the light chains. Here, like this, four are present. Let's see this one. That only the next sentence. Two of them are the long and the identical heavy chains. They are called as a H chain or identical heavy chains. And other two small chains are called as the L. That is represented as the formula like they are writing. That is the H2, L2. Heavy chains are 2 and the light chains are 2. That's why that is represented by H2 and L2. Next, the two chains are linked by which bonds? Already I said this one is a disulfide bonds. Linked by the disulfide bonds. One of the molecule is called as a FAB end. This molecule, this is called as a FAB end, which is the antigen with this antigen is attached. Next is the other end is called as a FC end, which may be attached to the B cells. Here, next, based on the structure, the antibodies are again based on the mobility we discussed. Based on the mobility, antibodies are two types are there. What are they? Circulating antibodies and as well as the surface antibodies. Here, the depending upon the mobility, antibodies are two types. Some are freely moving, and that's why we call as a circulating antibodies. They are present in the blood and lymph. The one more is there that is called as a the surface antibodies. These surface antibodies are present on the B cells. Here, this is about the next. Based on the structure, the antibodies are mainly the 5 types are there. The simply alphabetically IG. Every time we are writing IgA is the one type of the antibody. Next, IgD, IgE, IgG, and IgM. Like this, the antibodies are majorly there are 5 types are there depending upon the structure, depending upon the mobility, 2 types circulating and the surface. Depending upon the structure, there are 5 types are there. They are the IgA, IgD, IgE, IgG, and IgM. Here, this one is about the immunoglobulins are also called as a antibodies. Where they are present, the antibodies are present on the majorly the circulating means inside of the blood, lymph they are circulating, and sometimes it is attached to the B cells, surface of the B cells they are attached, and this one is mainly helpful for the entry of the antigens, prevent the entry of the antigens. If the antigens are entered, they are engulfed and destroy that. Here, this one is about the immunoglobulins. This one is a very, very important question. Among the B cells, and in this lesson, the 2 are the most important questions. First is the already we discussed, that is the B cells, and second one is the immunoglobulins. Here, the compulsorily you draw the diagram for this answer. Next, we go to the next SAQ. Next is the immunity. Here, the 2 types of immunities are there. What are the immunities we are going to discuss? The major here is the first, before the discussing of the types of immunity, one more question is there. Here it is the various types of barriers for the immunity. Here, for the immunity, which barriers are present? We are going to discuss. That also important question. See this one. Mainly the barriers of the immunity. Inborn, the resistance are having the mainly the disease, they causing the diseases, and the which one is protecting that one, that is called as a barriers. Barriers are mainly 4 types are there. They are called as a first physical barriers. Here, the physical barriers, which one is there means the skin and the mucous membrane. Skin is a protect from the antibodies. That's why they are called as a barriers. They protect from the antigens and microbes. That is the physical barriers are 2 types are there. The skin and the mucous membrane. How they are protect means skin prevent the entry of the microbes. Skin first only they prevent from the entry of the microbes. And what about the mucous membrane? Gelatinous substance is released that mainly kills the microbes that enter into the body. Clear? After the entering, for example, saliva, tears are having their mucus. That is mainly for after the entering of that, they are removed. That we call as a physical. They are present outside of the body. That's why we call as a physical barriers. Clear? Next is the second type is called as a physiological barriers. Clear? They are enter inside of the body. Inside of the stomach, HCl is produced. All of you know this. Digestive system time we discussed. The HCl is produced. That HCl only converts the pro enzymes into the active enzymes. Next is the what it is, the HCl. Here HCl is present inside of the stomach and the saliva in mouth. Here the tears of the eye. Three are there. What are they? I'm writing here. See the physiological barriers. Three are act as a physiological barriers. The HCl, where it is inside of the stomach. Inside of the stomach, HCl is present, and the saliva and the tears. Here, mainly the saliva and tears, both of them are having the enzyme is there. That is called as a the lysozyme enzyme is there. Here, lysozyme enzyme is there. With the help of the lysozyme enzyme, they kills the bacteria. See this one, the secretions like HCl of the stomach, saliva of the mouth, and the tears from the eyes, the barriers against the microbes. Saliva and tears, both of them are having the, see this one here, having the saliva and tears have the lysozyme which kills the bacteria. That is about the physiological barriers. Means the which one is act as a physiological barrier? HCl in the stomach and the saliva in the mouth and the tears from the eyes. Here they are which one? Tears and the saliva, both of them are having the enzyme that we call as a lysozyme enzyme. This lysozyme enzyme helpful for the destroying of the microbes. Next is the cellular barriers. Here the cells, some cells are also fight against the antigens that we call as a cellular barriers. The cellular barriers are mainly the neutrophils. Here, which one? Neutrophils. NK cells means natural killer cells. Here, neutrophils, natural killer cells, and the monocytes. Monocytes. These are act as a cellular barriers. Here, the neutrophils are the one type of the lymphocytes. Natural killer cells also, they are modified the lymphocytes only. Next, the monocytes. These all are taking the inside the antigen and destroy. That's why we call it as a cellular barriers. Cells are destroy the antigens. That's why we call it as a cellular barriers. The neutrophils, monocytes, natural killer cells in the blood, and the macrophages also. Macrophages are the cellular barriers. They engulf or kill the microbes. Here, this one is about the cellular barriers. Next, the cytotoxic or toxic the barriers. They are called as a cytokinins to protect the non-infected cells from the further infection. Listen here, cytotoxic barriers. What it is means what is the cytotoxic barriers means? They are protect the nearby beside the which the affected cells are there. For example, this one is the infected cell. Simply we imagine like this is the infective cell. Surrounding of that number of cells are there. Only the one cell is affected. The surrounding cells are already healthy only. They are not affected by the microbes. That protected by this cytokinins. Here, cytokinins protect the non-infected cells from the further infection. Means the infection is present inside of this only. They are not infected to the other cells. For that purpose, cytotoxic barriers are helpful. Here, like this, the three types of the four types of barriers are there. What are they? Physical, physiological, cellular, and the cytotoxic barriers. This is about the types of barriers. Next, we are going to discuss about the humoral immunity. Here, that immunity is mainly two types are there. They are called as a, first one is called as a humoral immunity. And the cytotoxic or cellular immunity. Cytotoxic or cellular immunity. Here, first we are going to discuss about the humoral immunity. Mainly humoral immunity occur in the the antigens are present in the outside. Outside means exo antigens are they are killed by the humoral immunity. See this one. The immunity is mediated. Immunity mediated by the antibodies released into the fluids. Here, their plasma and lymph. This is mainly extracellular. Extracellular antigens are destroyed by this humoral immunity. This immunity is by the antibodies only. In this, the immunity, which one is mainly take a role means the antibodies only kills the antigens. Here, humoral immunity time, humorals means the blood or lymph. Fluids only, inside of the fluids only, the antibodies are present, that antibodies are kills the antigen. That's why we call it, the name is there, that is called as a humoral. Humors means the blood and lymph liquids are called as humors. Here, next, the immunity mediated by the antibodies released in the fluid is called as a humoral immunity. Here, next, these antibodies are present on the surface of the B cells. Here, on the surface of the B cells, I'll draw the diagram clearly. See this one. This one is the B cells. You know the diagram about the B cell. B cell is having the outer side, the antibody surface. Antibodies are present like this. This one every time B cell means draw the diagram like this only. This one is a B cell. Normally, the B cells here, the antibodies are present on the surface of the B cell. Here, where they are present, which one is kill the antigens. The antibodies are kills the antigens. They are present on the B cell. Here, on the surface of the B cell, the antibodies are present. These antibodies, see, here the antibodies present on the B cells. Antigens get attached to the FAB end of the antibody. Already B cells, FAB end means this side. FAB end, any antigen through the blood circulation or the lymph circulation, they enter to the, where it is present, they are antigen binding site. Which one? FAB fragment side, the antigen is present. See this one? The antigen gets attached to the FA end of the antibody. The B cells engulf the antigen and they get processed here. They take this one and they get processed here. Next, the antigenic fragments are displayed with the help of the MHC2. Here the MHC2. Major histocompatibility, two cells are there. These cells are first, the B cells are taking the antigen where by the FAB. Here on the B cells, the antibodies are present. These antibodies are taken the antigen, they are destroyed into the fragments. That fragments are identified by the MHC2. Here, that immediately the MHC2 give the information to the that is identified by the T helper cells. The complex here it is formed that is identified by the T helper cells. T helper cells are producing the secretion interleukins. Here there are some chemicals they are released that we call the interleukins. Immediately the interleukins are released. The B cells are differentiated into two types of cells. One is called as a plasma cell. One type of cell is called as a plasma cell. Another is called as the memory cell. Memory cell. Here the first any antigen is enter inside of the body, the humoral means through the circulation they enter to the near the B cells, the antibodies. Here antibodies are taken this one and make it as a fragments, antigenic fragments that is first taken by the MHC2. The complex is formed immediately. The T helper cells are identifying this one and immediately they are releasing the substances. They are called as interleukins. Interleukins act on the B cells. The B cells are differentiated into two types. They are called as plasma cells and the memory cells. What is the function of plasma cells? In the B cells only we discussed. Plasma cells are simply, they are releasing the antibodies. Antibodies are released by plasma cells. What about the memory cells? They store the memory about the antigen, means the long term memory cells are stored inside of the body. If same antigen enter in our body, that time they protect the body. Clear? This one is about the memory cells. This is about the tumor. See this one. The T helper cells react with the antigen complex. Here it is a complex is formed now. This antigen complex is identified by the see here the T helper cells. Here TH cells react with the antigen MHC2 complex to release the interleukins. Right, I said now interleukins are released. The interleukins stimulate the B cells to produce the memory cells and the plasma cells. Clear? What about the memory cells? Memory cells store the information and produce the quick response if any second time they enter inside of the body future. Here, next is the plasma cells. What is the function of plasma cells? Explain here. Plasma cells releasing specific antibodies into the plasma and extracellular fluid. Here, this is about the function of the plasma cells. Next, the antibodies opsonize and the cross-linking of the antigens leading to the agglutination of the antigen and antibodies. Here, next is they are participate the soluble antigen. They also activate the phagocytes and the complement systems. Clear? This one is about the means the simply humoral immunity means which one is response? The antibodies are response. Antibodies are act on the antigen. That is called as also the antigens are present in the humorals like blood and the lymph. That's why we call it humoral immunity. In this humoral immunity, which one is participating? Mainly the antibodies are participating. Antibodies are participating in the humoral immunity, not the cells. Only the fragments, antibodies are participating in this one. That's why we call it as the humoral immunity. Clear? That is, which one is involved in this one? Humoral immunity. B cells are involved. But next, the coming is called as cytotoxic immunity, or also called as cellular immunity. In this, which one they are not participating? B cells are not participating in the cell-mediated immunity, or also called as cytotoxic immunity. Clear? Now, next immunity, next type of immunity we are going to discuss. Next, the second type of immunity. Next is the cell-mediated immunity. Second type of immunity, that is called the cell mediated or cytotoxic immunity. What it is means, inside of the cell they are occur. Before that, humorals are having the antigen, but here they are endogenous. Before that, exogenous antigens. How they are means the humoral immunity. Clear? Exogenous antigens. Exogenous antigens destroyed by which immunity? Humoral immunity. Humoral immunity. They destroyed by the humoral immunity. What about the endogenous? They enter inside of the cell. That time, which is how they are destroyed means endogenous. Endogenous antigens are destroyed by the cell-mediated immunity. Cell-mediated immunity. Yeah. The exogenous means they are outside. Outside means the inside of the blood or plasma. They are lymph. They are present. That is destroyed by that antigens are destroyed by the humoral immunity. They are the antigens are enter inside of the cell. That we call as the endogenous. Enter inside. That's why we call as the endogenous antigens. This is destroyed by the immunity. That we call as a cell-mediated immunity. Now we are going to discuss about the humoral immunity. Already we discussed. In that, they are, which one is participating? The B cells are participating in this one. In the cell-mediated immunity, the T cells are participating. Here, majorly T cells are participating. The B cells, T cells, already I said. Which one? That is the one type of lymphocytes. Beta lymphocytes are also called as a B cells. T lymphocytes are also called as a T cells. In the cell-mediated immunity, T cells are mainly participating. See here, observe here, the cell-mediated immunity, the immunity mediated by activated T cells. Already I said, activated T cells are the natural killer cells. NK cells are participating in this. That's why it is called as a cell-mediated immunity. In this, the cells are involved. Before that, B cells are involved, but B cell directly cells are not involved in this one. B cells are producing the antibodies. That is antibodies that are participating. But here directly the T cells are NK cells are participating. That's why we call as a cell-mediated immunity. Here, the antigen, where it is antigen presenting cells, process the exogenous antigen. For example, in exogenous is there, that is altered as a altered. The cells are formed. I mean, they are formed as an endogenous. Here, alter cells. The process endogenous antigens. Here, these cells display the fragments on the plasma membrane. They are display the fragments on the plasma membrane. Next, then ASP means self-altered the cells. Next is the APC means antigen presenting cells. Here, ASC means self-altered cells. The APCs means every time the antigen presenting cells. See here, T cells recognize which one? Antigen presenting cells they can recognize, as well as self-altering cells also they can recognize and bind to them. Here, where they are present, the T cells are bind to them. Then the activated T cells and the normal memory T cells are formed. They are T helper cells, T cells attached with these two, automatically what will happen? They are transmit as a T cells are transformed it as a B cells are formed as a plasma cells, memory cells. Now, like that only the T cells are formed it as a the two types. They are the T activated cell, and the memory cell. Same like a B cells, that is also having the memory cells. See, observe here, they are T cells, activated T cells are formed and the memory cells are formed. Here, the two types, activated T cells or T activated cells and the memory cells are formed. What is the T activated cells are formed means activated T helper cells produce the, means they are producing the interleukins. Here, the two are formed. Which one? Activated T cells and memory cells. Activated T helper cells, they are producing the some substance same like the B lymphocytes they are producing now. Like that only here they are producing the interleukins. Interleukins. Here the interleukins, the transform the activated T cell into the effector cytotoxic cell. Immediately, the T activated cell in the presence of the interleukin cell, CTC is formed. Here, what is the CTL is formed. CTL is formed. See this one. What is the CTL means? The cytotoxic, the T lymphocytes is the cytotoxic T lymphocytes. Which one? The T activated cell, immediately they are releasing the interleukins. The interleukins are formed as a cytotoxic, the T cells are formed, T lymphocytes are formed. That's simply we call as a CTL. Here, next the CTL attached to the, where the infected cell is there, that infected cell is, which one is attached? CTL. CTL is attached to the infected cell and they make it as a, I'll write a diagram and then you can understand easily. Wait. The interleukins are released automatically. Interleukins are released. That interleukins are transform the T helper cells. The T helper cells are releasing the which one? Interleukins. Here, interleukins are released. With the help of the interleukins, they are formed as a CTL. Here see this. CTL means cytotoxic T lymphocytes or killer cells are formed. These killer cells, they are what it is? They are attached to the infected cell. These cells are there now. They attach to the infected cell. This one is the infected cell. Infected cell. Here, they attach to the release the porphyrins, granzymes. Here, they are attached to the infected cell. They are attached to the infected cells and releasing the two substances. They are called as a porphyrins and granzymes. Porphyrins and the granzymes. When the TCL is formed, that immediately attach with the infected cell and the after attaching of the infected cell, they are releasing the two types of substances. One is called as a porphyrins and the second is called as the granzymes. What is the function of the porphyrins means? They make the holes on the infected cell, which is having the microbe. That cell becomes as a having the number of opening spores are occur. That by the porphyrins. Porphyrins makes the holes on the infected cells. And what about the granzymes means? After the formation of the opening, granzymes are enter inside of the infected cell. After that, what will happen means this infected cell becomes as a larger and becomes as a swelling and they are destroyed. That we call as a apoptosis. That condition is called as a apoptosis. Say first, the active T helper cells are formed as a first CTL cells that is attached to the infected cell. Immediately they are releasing the two substances. One is called as a porphyrin, second is called as a granzymes. The parfenins are helpful for the make the holes in the infected cells, and the granzymes are the liquid-like structures. They are enter inside of the cell, infected cell. The cell becomes as a swollen, and they destroy. That process is called as a apoptosis. Here I will explain in the sentences. So wait. See. Here the activated T helper cells produce the interleukins. Interleukins transform and activate the T cells into the cytotoxic T lymphocytes. CTL cells are also called as a killer cells. That CTL cells attach to the infected cells and releasing two substances. One is called as a parferin, second is called as a granzymes. Parferins produce the pores in the plasma membrane. In the plasma membrane, they make the holes of the infected cells. And what about the granzymes? The granzymes enter into the cytoplasm of the infected cell through the pores, and the proteins. That means they are enter some substances. The activating of the proteins destroy the infected cells. That process is called as a apoptosis. Clear? This is the natural killer cells are also similar action. Natural killer cells directly they can take the infected cell and that is destroyed. Same like a CTL cells. Clear? This one is about the cell mediated immunity. Simply the humoral immunity means they are, which is performing the humoral immunity means the antigens are present in the humors, means antigens circulating antigens are undergoes the humoral immunity. The inside of the cell they enter, that is undergoes the cell mediated. Exogenous antigens are undergoes the humoral immunity. Endogenous antigens are undergoes the cell mediated immunity. Here with this one is about the immunity, two types of immunity which is completed. Next one more question is there, that is the difference between the HIV and AIDS. Tomorrow we continue with this. In this immunity, the only one SAQ left. Next we'll meet tomorrow same this time. 1 second.